All right, now we'll look specifically at carbohydrate metabolism. So carbohydrate metabolism, again, basically breaking down carbohydrates all the way to glucose and then breaking down glucose to generate ATP molecules, which are then used by the body for energy. Carbohydrate metabolism occurs inside of the cell, primarily at the site of the liver, muscle, brain, and heart. Fructose and galactose, again, I mentioned this in the metabolism lecture, but these can enter into the glycolysis pathway, but often they will be transported to the liver to be converted to glucose, and then glucose will travel to the specific tissue or cell to be metabolized into ATP. So most carbohydrate metabolism, we're really talking about glucose metabolism. So the processes of carbohydrate metabolism, we're going to talk through glycolysis. We're going to talk about pyruvate and acetyl-CoA and lactic acid. We're going to talk about the Krebs cycle and the electron transport chain. Here's a diagrammatic overview of that. So glucose through glycolysis is converted to pyruvate. Pyruvate, in the absence of oxygen, is going to be converted to lactate. Lactate will actually go to the liver to be converted back to glucose and then can come back to the cell when oxygen is present and will then just re-enter glycolysis and create pyruvate anew, and then will be converted. Pyruvate will enter into the mitochondria and will be converted to acetyl-CoA. Acetyl-CoA will undergo the Krebs cycle, and then ultimately the coenzymes from the Krebs cycle will pass through the electron transport chain, and ultimately in the process, the final process of oxidative phosphorylation, we're going to create ATP molecules. Just another diagrammatic overview. All right, so let's talk through each part individually. So glycolysis, ultimately, we're converting one glucose molecule to two pyruvate molecules. And I like this diagram because it shows us the chemical form, sort of these molecules. So glucose, remember, is a 6-carbon molecule, a hexose. And then it makes sense then, pyruvates are 3-carbon molecules. So it makes sense we can take one glucose, 6 carbons, and make 2 pyruvate, which are each only 3 carbons. This isn't showing it, this is kind of showing it, yeah, that dephosphorylation step that I told you I would point out to you. So glyceraldehyde 3-phosphate has the phosphate group, and so in a dephosphorylation, ADP is actually going to harness that phosphate group away from glyceraldehyde 3-phosphate and thus make pyruvate. So glyceraldehyde 3-phosphate is dephosphorylized to make pyruvate. And then similarly, the ADP molecule is phosphorylated because it's gaining the phosphate group. We also see a redox reaction. The NAD is reduced to, NAD plus, is reduced to NADH. It gains an electron. And so technically the glyceraldehyde 3-phosphate is oxidized to pyruvate. Excuse me. This whole process is itself anaerobic, so we can undergo basic glycolysis without the presence of oxygen, but we're not making a whole lot of ATP in doing that, only 2 molecules. This is just showing glucose converted to pyruvate, and also where glycogen might play in. It's also showing where galactose and fructose would play into the overall pathway. So in some cases, galactose and fructose would be converted to glucose, like I said, and just enter this way, but they can also enter directly into the pathway. And again, this isn't even the entire process, but this is just showing some of the intermediary reactions that take glucose all the way down to make pyruvate. And again, you see actually a few times this phosphorylation, dephosphorylation. So at this step, this molecule has 2 phosphate groups, and by the next step, it only has 1 phosphate group. You see a dehydration synthesis. All right, so then pyruvate has multiple fates. We're going to talk primarily about these two, but pyruvate, again, in the presence of oxygen, so, like normal steady state activity, will convert pyruvate to acetyl-CoA. If we are like running our fastest marathon time, or our fastest 5K time, we're actually going to see some pyruvate is going to be sort of shunted to make lactate because we don't have enough oxygen to convert all the pyruvate we're making into acetyl-CoA. These other two molecules are, the oxaloacetate is important because it's important to know that, and again, we'll look at this in subsequent slides too, but oxaloacetate can be made by pyruvate. Otherwise, it is typically regenerated in the Krebs cycle, but it's important to know that if we are continually having acetyl-CoA coming in through the Krebs cycle, we do need to have another way to also make oxaloacetate. So it's important to know that we can make it from pyruvate. And then in some cases, this is actually a transamination process if we don't really need pyruvate, if we don't need to make a lot of energy, or if we have enough energy, we can actually use pyruvate to make the amino acid alanine. So just looking at what happens to lactate, or lactic acid. So if we're here in the site of the muscle, and again, we're running a really fast 5 miles or something, we may be converting so much glucose to pyruvate that we don't have enough oxygen to convert all that pyruvate to acetyl-CoA. So we will convert some of the pyruvate to lactate for the time being. It's just kind of like a holding place. Lactate will move through the bloodstream to the liver, where the liver will convert the lactate back to glucose, and then maybe you would store that as glycogen, but might actually just send the glucose back to the muscle. Say once the five mile run is over, you're still breathing hard for a little bit, right? You kind of have a period of time where your body cools down. It's really kind of in that cool down period, if you built up lactate, that the body would convert all that lactate back to glucose and then utilize the glucose either in the liver or in the muscle tissue. But most of the lactate, for the most part, the lactate will be gone within 45 minutes after the activity has kind of come back to a more casual pace. So this idea that, like, if you ran a marathon and the next day you're really stiff, that you have a lot of lactic acid in your muscles, is really not actually physiologically true. The Krebs cycle. So again, we're following the acetyl-CoA molecule. The Krebs cycle is basically just a series of, it's kind of like a revolving door of reactions. And it's not quite, although it kind of looks like it, it's not quite that the acetyl-CoA just passes through all these different reactions. There are all these various molecules throughout the Krebs cycle that are really important to be present to allow these various reactions to happen. But ultimately, what I want you to understand about the Krebs cycle is that we need both oxaloacetate and acetyl-CoA plus all the other intermediaries, but we're not going to talk about them in this class, really. And in all these various reactions that are going on, we are creating a little bit of ATP, which is not shown in this slide. But we also see the presence of these coenzymes, which, if you remember from the basic metabolism lecture, I told you are B vitamin derivatives. And what I want you to recognize is that they are all undergoing reduction reactions where they're gaining electrons and gaining hydrogen atoms. And this is really important because what we'll see, let's see in the next slide, is the electron transport chain. So now we're deep inside the mitochondria. This is actually the outer mitochondrial membrane. This is an inner mitochondrial membrane. We're inside we're inside the matrix of the mitochondria. And we see these in this intermitochondrial membrane, we see these 1, 2, 3, 4 protein molecules in the membrane, the intermembrane. these are actually going to help us create what would be called a hydrogen ion concentration gradient. So you'll see the NADH, you'll see the FADH2. These are all basically going to now undergo oxidation reaction where they're going to give up their hydrogen atoms to start creating this hydrogen concentration gradient. So we're going to start to see all of these hydrogen molecules build up here in the intermembrane space. And then ultimately, we're going to have high hydrogen concentration. We're going to wind up with low hydrogen concentration here. We're going to see all the hydrogen atoms now shuttle back through what's called ATP synthase, and this is where we see oxidative phosphorylation, where we have a bunch of ADP molecules at the ready to now receive, and free phosphate molecules. They're now going to receive the hydrogen atom and thus generate ATP. The ADP will become phosphorylized to ATP. And this is where this is the molecule where we make all of our ATP. So just a little overview of the whole carbohydrate metabolism pathway. Glucose, one molecule of glucose is going to break down to make 2 pyruvate molecules, and then those pyruvates are going to be converted to acetyl-CoA. The acetyl-CoA is going to pass through the Krebs cycle, the electron transport chain, and ultimately, the acetyl-CoA will pass through the Krebs cycle, thus generate a bunch of coenzymes. The coenzymes will then donate their hydrogen atoms to the electron transport chain, and then all those hydrogen atoms will create a concentration gradient, and then pass through the ATP synthase molecule, thus creating ATP. So from one molecule of glucose, we can make roughly 30 to 38 molecules of ATP. We also do create carbon dioxide and water as byproducts. And then remember that just without oxygen, or without sufficient oxygen, pyruvate will temporarily be converted to lactate, which will then ultimately be converted back to glucose to make ATP. So one glucose molecule makes 30 to 38 ATP, but it's obviously there's a range. It varies a little bit per glucose molecule.